Okay, so welcome to my live stream looking at the biological membrane. So we've got a question here that's asking us about the fluid mosaic model and how it describes the plasma membrane of all living organisms. So the question says, how does the fluid mosaic model describe? So the first thing I would be doing is I would highlight, so it's the fluid mosaic model. How does it describe the structure of the plasma membrane? So it's important that when we look at these questions, we know what it is that they're asking us. So we need to remind ourselves about what does the phospholipid bilayer actually look like. So let's just have a quick look then at the phospholipid bilayer. So what we can see then is if we were to look at the phospholipid bilayer, we know that we have got our hydrophilic phosphate heads orientated on the outside and our hydrophobic tails orientated on the inside. So this has got a 2 marker to it. So therefore, what I'm going to say is one of the parts that describe it is this phospholipid bilayer. And then the second thing is the fact that those hydrophilic heads, which have our phosphate, are orientated on the outside, and the hydrophobic tails are on the inside. So we have the fact that our hydrophilic heads orientate on the outside and our hydrophobic tails orientate on the inside. So that is where we can see that we've got this phospholipid bilayer. But we have to remind ourselves that actually, if we're looking at this phospholipid bilayer and we're looking at the fluid mosaic model, what we need to understand is that we have proteins as well. So the proteins are embedded in the membrane. And those proteins are actually free to move, as are the phospholipids. Just correct that spelling there. So straight away, what we've actually noticed is that it's a fluid mosaic model. So the fluid is to do with the phospholipids and the proteins being free to move. So the fluid is they can both move, and the mosaic part of it is that those different proteins are actually embedded randomly with that structure. So I'm just going to change the color here so that will show up clearly that that's the answer. Okay, let's move on to the next question then. So the next question is saying that the plasma membranes are partially permeable. Well, the first thing that I would do is I would start again with my highlighting. What does that partially permeability actually mean? So when they talk about it being partially permeable, it means that those proteins are going to let some things through. And what we have to understand is that the phospholipid bilayer is going to be very specific that it is not going to allow anything that is polar. So polar, repelled. So these are important notes that I would tell myself when doing this. So let's see what the question's actually asking. So the plasma membrane are partially permeable, allowing some molecules to cross the membrane with relative ease. One molecule that crosses the membrane easily is the steroid hormone. Right, so they've given us a little hint here. They've told us it's a steroid hormone, which is produced in the ovaries from, oh look, cholesterol. Again, they've given us a bit of hint here. We know that the cholesterol is able, because we can see it here, the cholesterol is able to sit inside the phospholipid bilayer because it is a non-polar substance. So therefore, the fact that this steroid hormone, progesterone, is made from cholesterol means it would be non-polar too. So the first thing I would want to say is the fact that we've got the progesterone, if it can cross the membrane, has to be hydrophobic because it has the ability to go across. So hydrophobic progesterone And because it has this hydrophobic progesterone, it therefore means that it is able to diffuse straight through the phospholipid bilayer. Because it's able to get across through the fatty acid chain. So what we've got to understand is that it's not going to be repelled away. So name one other molecule that can cross the plasma membrane. Well, let's just have a look here. We know that we're talking about the movement of substances through the membrane. So what do we know? Well, we know that if it's a small molecule or a non-polar molecule like progesterone, aldosterone, so one other molecule that can cross the plasma membrane, well, for that one, we could say anything from the likes of our carbon dioxide, or we might want to say oxygen because these are small molecules. The other thing that we could actually say is we could say any of our hormones that we know are non-polar, so aldosterone is another example of a hormone that could get through. So then we look at the next question, and the next question is asking about active transport. And it's asking about active transport and the use of the ATP to pump the potassium ions through the cell surface membrane against the concentration gradient. So they've given us lots of hints here. We're going against a concentration gradient. It's active transport. ATP is provided. So whenever you see questions like this, I always would say use that as a guide for you making your revision notes, because what you actually have here is a definition of what active transport is. Active transport is where a substance, in this case, potassium ions, are going to be pumped against their concentration gradient. Now, they are ions, so that therefore allows me to identify that they are, for example, potassium K plus. Potassium is a charged particle, and that's why we're talking about it as an ion. So therefore, ATP is required to move it against its concentration gradient. But that's not what the question is actually asking us. What the question is actually asking us is, what is ATP? What is ATP made up from? So this is where we need to take our time and actually look at what is active transport and what is ATP. So let's have a look at these notes then. So we know that we've got adenosine triphosphate. So we know that we have got our phosphate group because they've told us in the question that there's the phosphate group. But what they want to know is what is the two other molecules. So they're ignoring this part. What they're looking for is, do you actually know what this and this is? So do you know that it is made from adenine? And do you know that it is made from a ribose sugar? So when you look at questions like this, it's important that you note what they are actually asking in the question. So let's move on. Let's have a look at the next question then. So here we've got information about the membrane. So a group of students investigated the effects of temperature. So straight away I can see the effects of temperature. So before I move on to look at the question any further, I would always tell myself, right, what does that actually mean? Temperature, what should I be noting? Well, temperature, I need to think about kinetic energy. Now we're talking about the membrane here because it's talking about the effects on the membrane. So I'm going to remind myself that membranes are fluid mosaic model. So because we're looking at the fluid mosaic model, we're looking at membranes, we're looking at the effects of temperature, kinetic energy. So all I'm doing right now is I'm giving myself a little hint as I do this question as to what might come up. Where might they want me to go with my knowledge? So let's look at what else they're telling us. So we've got a colorimeter, okay? So we're looking at quantitative data. So I've got my colorimeter and I've got my data being collected. Okay, so what are they telling me? Right, we've got a purple pigment that's going to leak out of the cells when it was exposed to different temperatures. Okay, so they've then told me what the temperatures are. So we can see that as we go down here, the temperature is increasing. They've taken 10 repeats. So the number of readings that they took was 10, and then they've told me that there's a mean absorbance. Okay, now they've asked me about that standard deviation. So they've said using the student's t-test formula, a formula they've given me, below, calculate the value of t between the data for the 50 and the 60. So what I would, again, always do is I'm going to use this information to help me to work this out. So what does XA mean? Well, they've told me that this is the mean, and I've been asked to do, I'm going to change color on my highlighter here. I've been asked to do 50 and 60. So therefore, I need these numbers and these numbers. So what do I need to do? Well, let's just add these numbers in then. So when I am looking at it, what have I got? I've got A, which is my 50, so 0.21 minus my B, which is 0.44. So all I'm doing is there's my A, there's my B. So then I'm going to divide that by the square root of, so the standard deviation of A is 0.06, and that is squared, divided by the sample number, which was 10, because they told me in the question. And the same again, 0.18 squared for B, divided by 10. So if I go through and I use my calculator for that, what that will tell me is that will give me an answer which is 3.83. So it's important when you are doing these questions that you take the time to really understand what it is that you are trying to show. So let's keep going with this question then and see what it says. What's the next part is asking me? Well, the next part is asking me is, do I actually understand what that is I've just worked out? So I've calculated the t value. Now, remember a t-test, whenever you are doing this, you want to remind yourself that a t-test is about calculating and comparing the means. So we're going to compare means of two sets of data. So we had those two means and we are now comparing them. We've got a number which is 3.83. Now it tells you in the question that you have got, I'm just going to change my color again. Let's go blue. A significance level, my significant level of 5%, which is the one we use in biology. It's given me my 18 degrees of freedom. How did they work that one out? Well, we had 10 plus 10, which gives us 20, minus 2 gives us the 18. So that's how they worked out the degrees of freedom. They're not expecting me to work that out. They've told me in the question. What they've then told me is they've told me in the question that when I read it off a table, that value would be 2.10. So now I need to work out whether the null hypothesis can be accepted or rejected. So a null hypothesis is that there is no significant difference between the mean absorbance of these two temperatures. So therefore, if I look at this then, what's the value I got? I calculated it as 3.83. Whereas the significance level of 5% gave me a critical value of 2.10. So because my number is greater than 2.10, I am going to reject the null hypothesis. So what I need to note is I'm going to reject the null hypothesis. Why am I going to reject the null hypothesis? Well, the reason I'm rejecting it is that my calculated value is greater than the critical value. So I'm using their terminology. So what does that actually mean? The fact that my value of 3.83 is greater than 2.10, what that means is that the difference is significant. So the difference between the means is significant. So that's important whenever I answer a question like that. Now, if I had actually got a number which was lower, so if I'd calculated the wrong answer to the one above and I got an answer which was less than 2.10, for example, if I'd have got a negative number, I would have needed to use that value, because what they will do is they will read your whole answer and they will double check what you've actually written, and you have to follow what is written that you calculated. So if I'd got a negative number, so let's just say, I'm going to make this up and we'll do it in blue. I'd got a negative 1.43. Let's just say if I got negative 1.43 as my calculated value, that would therefore be less than 2.10. So minus 1.43 would be less than the 2.10. Because it's less, I would accept the null hypothesis. And because I'm accepting the null hypothesis, it therefore means that the difference is not significant. So it's really important that you use the information and your calculated values. Okay, let's move on. And I'm going to finish with this question here. So students plotted the data onto a graph shown in figure 23. So we can see here that the absorbance is on my y-axis and the temperature is on my x-axis. So the next thing I need to do then is I need to decide how to answer this question. So again, I'm going to use my highlighter and I'm going to make sure that I have highlighted some keywords. So what is it actually asking me to do? It's asking me to describe, and then, so I'm going to use a different color here. It's asking me to explain. Describe the pattern and then explain the pattern. So it is very important that when you look at this, that you are actually looking at the information and the command questions. Describe the data. Well, as I increase the temperature, well, it doesn't really do anything, to be honest, at the beginning. So it's important that you are looking at this in the right way. So as I increase the temperature, nothing's happening for the first 20 degrees. So don't jump to a description that says, as I increase the temperature, the absorbance also increases. Well, no, it doesn't. So it's important that you're describing exactly what you see. So I've got low temperatures here and I've got no change. So there's no change in absorbance. I'm describing it. Now let's explain why. Why might there be no change? Well, because the membrane's intact. What have I got to remember? I've got to remember that I have my cholesterol. My cholesterol in my membrane is going to stabilize. Now, this is not expected in the question, but I always like to push it a bit further to explain why it is that that's the case. The membrane's intact at low temperatures because there's no change. So then what happens as I increase the temperature? Well, obviously we can see as I increase the temperature, what's going to happen is we're going to see quite clearly a change. So remember what I said before? I said before that temperature is going to have an effect on the kinetic energy. So if I'm increasing the temperature, what's going to happen? There is then a steep increase in absorbance. Why might that be the case? Well, remember, an increase in temperature is going to result in an increase in kinetic energy. If there's an increase in kinetic energy, what's that going to do? That's going to mean that there's going to be more leakage. That leakage out is because either we've got that the membrane has been damaged, the membrane might be damaged. It could be that if I go too hot, my proteins denature. So remember when you are given a question that says describe and explain, that you are ensuring you're giving both answers. Otherwise, you will not get the full marking points. So I hope you've enjoyed this little introduction to how I